Hello everyone. Welcome to Pragya Goyal's Biotechnology. The topic for today is structure and properties of DNA. We are going to divide this topic into two parts. So today we are going to start with the first part. I am Mrs. Pragya Goyal, Assistant Professor, Department of Biotechnology, IPS Academy. What is DNA or deoxyribonucleic acid? It is a thread-like chain of nucleotides carrying the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. DNA and RNA are nucleic acids, alongside proteins, lipids, and complex carbohydrates. They are one of the four major types of macromolecules that are essential for all known forms of life. Most DNA molecules consist of two biopolymer strands coiled around each other to form a double helix. DNA stores biological information that is passed on from parents to children, providing instructions for how and when to make the many proteins needed to build and maintain functioning cells, tissues, and organisms. The DNA backbone is resistant to cleavage, and both strands of the double-stranded structure store the same biological information. This information is replicated as and when the two strands separate. A large part of DNA is non-coding, meaning that these sections do not provide information for protein structures. Within eukaryotic cells, DNA is organized into long structures called chromosomes. During cell division, these chromosomes are duplicated in the process of DNA replication, providing each cell its own complete set of chromosomes. Eukaryotic organisms like animals, plants, fungi, and protists store most of their DNA inside the cell nucleus and some of their DNA in organelles such as mitochondria or chloroplast. In contrast, prokaryotes like bacteria and archaea store their DNA only in the cytoplasm. Within the eukaryotic chromosomes, chromatin proteins such as histones compact and organize DNA. These compact structures guide the interaction between DNA and other proteins, helping control which parts of the DNA are transcribed. How do we know so much about DNA? Many scientists have worked through decades, through centuries, to provide us the information that we have today. The first piece of the puzzle was in 1869. This was the landmark year in genetic research because it was the year in which Swiss chemist Friedrich Miescher first identified what he called nuclein inside the nuclei of human white blood cells. The term nuclein was later changed to nucleic acid. He was originally trying to study the composition of lymphoid cells. The main source of these cells were pus-coated bandages collected from a nearby medical clinic. Instead, he isolated a new molecule he called nuclein, which is, as we know, DNA associated with proteins from a cell nucleus. In 1881, Albrecht Kossel identified nuclein as a nucleic acid and provided its chemical name deoxyribonucleic acid. He also isolated the 5 nucleotide bases that are building blocks of DNA and RNA: adenine, cytosine, guanine, thymine, and uracil. His work was rewarded in 1910 when he received the Nobel Prize in Physiology or Medicine. In 1882, shortly after Kossel's findings, Walther Fleming devoted research and time to cytology, which is a study of chromosomes. He discovered mitosis in 1882 when he was the first biologist to execute a wholly systematic study of the division of chromosomes. His observations that chromosomes double is significant to the later discovered theory of inheritance. In early 1900s, Theodore Boveri and Walter Sutton They were working independently on what's now known as the Bevery-Sutton chromosome theory or the chromosomal theory of inheritance. These findings are fundamental in our understanding of how chromosomes carry genetic material and pass it down from one generation to the next. In 1909, Phoebus Levine identified the base sugar and phosphate nucleotide unit of the RNA. Later, Levine identified deoxyribose sugar. Levine suggested that DNA consisted of a string of four nucleotide units linked together through the phosphate groups, which is known as the tetranucleotide hypothesis. He thought the chain was short and the bases repeated in a fixed order. In 1928, Frederick Griffith in his experiment discovered that traits of the smooth form of pneumococcus could be transferred to the rough form of the same bacteria by mixing killed smooth bacteria with the live rough forms. This system provided the first clear suggestion that DNA carries genetic information. In 1937, William Astbury produced the first X-ray diffraction patterns that showed that DNA had a regular structure. In 1943, Oswald Avery, along with his co-workers Colin MacLeod and Macklin McCarty, identified DNA as the transforming principle, supporting Griffith's suggestion. This is known as the Avery-MacLeod-McCarty. In late 1940s, Irwin Chargaff discovered that DNA is responsible for heredity and that it varies between species. His discoveries, known as Chargaff's rules, proved that guanine and cytosine units, as well as adenine and thymine units, were the same in double-stranded DNA. And he also discovered that DNA varies among species. In the late 1940s, Barbara McClintock discovered the mobility of genes, ultimately challenging virtually everything that was once thought to be. Her discovery of the jumping gene, or the idea that genes can move on a chromosome, earned her the Nobel Prize in Physiology. In 1952, DNA's role in heredity was confirmed when Alfred Hershey and Martha Chase in the Hershey-Chase experiment showed that DNA is the genetic material of the Enterobacteria phage T2. Late in 1951, Francis Crick started working with James Watson at the Cavendish Laboratory within the University of Cambridge. In May 1952, Raymond Gosling, a graduate student working under the supervision of Rosalind Franklin, took an X-ray diffraction image at high hydration levels of DNA. This photo was given to Watson and Crick by Maurice Wilkins and was critical to their obtaining the correct structure of DNA. Franklin told Crick and Watson that the backbones had to be on the outside. Before then, Linus Pauling and Watson and Crick had Arrhenius models with the chains inside and the bases pointing outwards. Her identification of the space group for DNA crystals revealed to Crick that the two DNA strands were anti-parallel. In February 1953, Watson and Crick completed their model, which is now accepted as the first correct model of the double helix of DNA. In 1962, after Franklin's death, Watson, Crick and Wilkins jointly received the Nobel Prize in Physiology or Medicine for their contribution to understanding of structure of DNA. Now, we are going to talk about the structure of DNA. DNA is a long polymer made from repeating units called nucleotides. The structure of DNA is dynamic along its length, being capable of coiling into tight loops and other shapes. In all species, it is composed of two helical chains bound to each other by hydrogen bonds. Both chains are coiled around the same axis. The length or pitch of one complete turn is 34 angstroms or 3.4 nanometers. One turn is the distance between two successive major groove and minor groove. In one complete turn of DNA, 10 base pairs are present. Therefore, the distance between two successive base pairs is 3.4 angstroms or 0.34 nanometers. The pair of chains has a radius of 10 angstroms, that is 1 nanometer, or a diameter of 20 angstroms. It remains constant throughout DNA. Although each individual nucleotide repeating unit is very small, DNA polymers can be very large molecules containing millions to hundreds of millions of nucleotides. For example, the DNA in the largest human chromosome, chromosome number 1, consists of approximately 220 million base pairs, and what will happen if you straighten it? It will be about 85 millimeters long, that is 8.5 centimeters. To talk about the monomer of DNA, that is nucleotide. In living organisms, DNA does not exist as a single molecule, but instead as a pair of molecules that are held tightly together. These two long strands entwine like vines in the shape of a double helix. The nucleotide contains both a segment of the backbone of the molecule, which holds the chain together, and a nucleobase, which interacts with the other DNA strand in the helix. A nucleobase linked to a sugar is called a nucleoside, and a base linked to a sugar and one or more phosphate groups is called a nucleotide. A polymer comprising multiple linked nucleotides, as in DNA, is called a polynucleotide. As we can see in the diagram, the sugar is the pentose sugar. At a position, we also have the phosphate group attached to it, and along with it, the other position, we have the nitrogenous base or the nucleobase adenine attached to the sugar. So sugar plus nitrogenous base is known as nucleoside, and all three put together is known as nucleotide. About the components of nucleotide in detail. The first component we are going to talk about is nucleobase or nitrogenous base. It is an organic molecule with a nitrogen atom that has the chemical properties of a base. A nitrogenous base owes its basic properties to the lone pair of electrons of a nitrogen atom. Nitrogen bases are typically classified as the derivatives of two parent compounds, that is pyrimidine and purine. These structures can be seen in the image. Purines consist of two ring structures, that is a 6-membered and a 5-membered nitrogen containing ring fused together. The examples of purines commonly found are adenine and guanine. Adenine's chemical name is 6-aminopurine and guanine is 2-amino-6-oxypurine. Pyrimidines have only one ring, which is a 6-membered nitrogen containing ring. Examples of pyrimidines commonly found are uracil, that is 2,4-dioxypyrimidine, thiamine 2,4-dioxy-5-methyl-pyrimidine, cytosine 2-oxy-4-aminopyrimidine. As we know, thiamine and cytosine pyrimidines are found in DNA, while in RNA, thiamine is replaced by uracil. Next component of a nucleotide is the sugar. The 5 carbon sugars, ribose and deoxyribose, are important components of nucleotides, and they are found in RNA and DNA respectively. This gives genetic molecules their name. The full name, as we know, of DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Both these sugars are pentose sugars. Earlier we saw that the combination of a base and sugar is called a nucleoside. What is the difference between the ribose sugar and the deoxyribose sugar? Ribose sugar, which we just saw, that is found in RNA, is a normal sugar with one oxygen atom attached to each carbon atom. In deoxyribose sugar, which is found in DNA, it is a modified sugar lacking one oxygen atom, hence the name deoxy. The difference of one oxygen atom is important for the enzymes that recognize DNA and RNA because it allows these two molecules to be easily distinguished from organisms. As we can see, this difference in the presence or absence of oxygen is at the 2' position of the pentose sugar. If a sugar, either ribose or 2-deoxyribose, is added to a nitrogen base, the resulting compound is called a nucleoside. Carbon 1' of the sugar is attached to nitrogen 9 of a purine base or to nitrogen 1 of a pyrimidine base by glycosidic bond. The names of purine nucleosides end in ocene and names of pyrimidine nucleosides end in idine. Example of purine nucleosides are adenosine and guanosine, while example of pyrimidine nucleosides are uridine, thiamidine and cytidine. Generally, when we name organic compounds, we use numberings from 1 onwards. However, in the case of sugar, we have used the denominations, that is 1', 2', 3', 4' and 5'. This is done so the positions can be differentiated from the nitrogenous base, which is also having carbon at different positions. To differentiate these two, the carbon in the sugar is numbered as a prime number. The next compound is the phosphate. Adding one or more phosphates to the sugar portion of a nucleoside results in a nucleotide. Generally, the phosphate is an ester linkage to carbon 5' of the sugar. They then form a bond which becomes the sugar phosphate backbone. This structure adds rigidity to the structure as the covalent bonds they form are much stronger than the hydrogen bonds between the two strands. In case of DNA, to indicate that the sugar is 2' deoxyribose, a small d is placed before the name. Example, AMP, that is adenosine monophosphate, CDP, that is cytidine diphosphate. These two belong to RNA due to the absence of a small d in front of their abbreviations. The next two examples are of DNA, that is DGTP, that is deoxyguanosine triphosphate, or DTTP, deoxythiamidine triphosphate. As can be seen from the diagram or the image, we can see presence of 1, 2 or 3 phosphate groups in the. Next we come to the bond which holds these nucleotides together. This is known as the phosphodiester bond. Inside the cell, a polynucleotide is built from free nucleotides that have 3 phosphate groups attached to the 5 carbon of their sugar. These nucleotides are thus called nucleotide triphosphates. During the formation of phosphodiester bonds, 2 phosphates are lost, leaving the nucleotide with 1 phosphate group that is attached to the 5' carbon by an ester bond. The second ester bond is formed between the 5' phosphate molecule of the nucleotide and the 3' hydroxyl group of the sugar in another nucleotide. Hence the name 3', 5' phosphodiester bond. The phosphodiester bonds in a polynucleotide chain form an alternating pattern of sugar and phosphate residues called sugar phosphate backbone. Phosphodiester bonds impart directionality to a polynucleotide chain. The polynucleotide chain has a free 5' phosphate group at one end and a free 3' hydroxyl group at the other. These ends are referred to as 5' end and 3' end respectively. The directionality of nucleic acids is essential for DNA replication and RNA synthesis. From the diagram, the first image, we can observe that the phosphodiester bond is formed between the 3' carbon and the 5' carbon of the next nucleotide. There are two ester bonds involved, one at the 3' position and the other at the 5' position. Hence, the name is known as the phosphodiester bond. Next, we come to base pairs. We have talked about 4 base pairs in DNA and 4 base pairs in RNA. How are these base pairs formed, and more importantly, why these only? These are the rules of base pairing or nucleotide pairing. The rules of base pairing are that A pairs with T, that is adenine pairs with thymine, C pairs with G, that is cytosine, which is a pyrimidine, pairs with guanine, which is a purine. In both cases, we are able to observe that the bond is formed between one purine and one pyrimidine molecule. Why is it like that? Why cannot two purines or two pyrimidines form the bond? This is because there is not enough space for two purines to fit within the helix, as the diameter is just 20 angstroms. But there is too much space for two pyrimidines to get close together for each other to form a hydrogen bond between them, because pyrimidine molecules, being single ringed, are smaller in size. So this solves the query that why does purine pair with pyrimidine, but why adenine cannot pair with cytosine or guanine with thymine? The answer is only A and T and C and G, there are opportunities to establish hydrogen bonds, shown here as dotted lines between them. We are able to see 2 dotted lines between adenine and thymine and 3 dotted lines between cytosine and guanine, indicating the presence of 2 hydrogen bond between thymine and adenine and 3 hydrogen bond between cytosine and guanine. These are the base pairing rules. Chargaff's rules came about. As we saw earlier, these rules were given by Erwin Chargaff in the late 1940s. These base pair relationships, we study them under 2 rules. The first rule is, in a DNA double helix, total purine will be equal to total pyrimidine, that is, there will be an equal amount of these two. It is actually right logically too, because if there is a purine, there will be a pyrimidine too in the complementary strand to bind them. This is an obvious consequence of the base pairing nature of the DNA double helix. The rules of base pairing tell us that if we can read the sequence of nucleotides on one strand of DNA, we can immediately deduce the complementary sequence on the other strand. The rule explains that whatever the amount of adenine in the DNA of an organism, the amount of thymine will be the same. Similarly, whatever the amount of guanine, the amount of cytosine will be the same. Therefore, to sum up the first rule, purine equals to pyrimidine, A plus G equals to C plus T, that is, purine equals to pyrimidine. Also, A equals to T, amount of A equals to T and amount of G equals to C due to the complementary nature of DNA, and A plus G upon C plus T, that is purine upon pyrimidine, equals to 1. The second rule states that the composition of DNA in terms of the relative amounts of A, C, G and T bases varies from one species to another, but is constant within a species. This molecular diversity added evidence that DNA could be the genetic material. To the end of this session, where we talked about various aspects of the structure of DNA. We will be continuing with more on structure and properties of DNA in part 2 in the next video. Thank you.